I'm Dr. Joseph Cobbs, and I'm here with Mr. Rick, and uh, we're gonna talk about a lab that I did, and it's a really great lab that we came up with. Uh, I think it really helps the kids to work together, collaborate, and to open their eyes up to a whole new world. Um, we Mr. Rick collected some pond water in mud puddles after the rains right by our school. And we also collected cheek cells inside our mouth, and we let the kids look at those and draw those and observe those. Before I started the lab, we discussed a little bit about it. I had actually made some drawings myself and showed the kids, and then also drew them on the board and told them what to look for to make it a little easier for them to find them, to know what to do. And then we had to go over how to use the microscopes, coarse focus, a fine focus, how to move the stage, all those different things. Okay. Yeah. So what did you think of the recording of the lab?
Well, I mean, it's always a fun lab. As you say, it opens up a whole new world to, I mean, where would you ever dream that you would see such interesting things from a puddle that you splash through and full of dirt usually and something to ignore and step around, but life is everywhere. To to use the imagination and start to think about that a lot of biology is very small and happening right below your nose, and you don't know it or don't see it, and that there is a whole world below the senses that you can't detect, and that if we extend our senses with these microscopes and other things, then it really does wake you up a little bit to what's really going on, and in so many ways. I think just the surprise element, it's just so fun to see their reactions, and what's that? Look, it just went flying by. It's like things are moving, things are happening, and they're on their feet. They're moving around, they're talking to each other, they're working together. All kinds of good things are happening. They're very involved and interested, and you're engaged. You're stepping in and making sure that they've refocused or moved the objectives to another level. Teaching them to move up and down and the focus within the drop. It gives them a sense of 3 dimensions, depth. They start to see things that they've never thought about before. So I'm sure I can say more, but let's turn it back to you and see what you have to say about it.
Well, I'm not as experienced as you are in doing these labs, and I find it a little more challenging, with my skills on using the microscope and just trying to get organized 18 kids to all be able to see something and to record it. We finally had to incorporate technology because we only had 3 or 4 scopes and that many kids. So they didn't all have time to look at the microscope and draw it. So a lot of times we were just having to make recordings with their phones, either recordings or taking pictures of it. And that helped them to have the time to really draw it. So that was something that I kind of did on the fly while we were doing it. I didn't realize when I started the lab that they were going to have difficulty making the drawings because there's 18 kids and 3 or 4 microscopes.
Yeah.
So that was an incorporating technology that I think was an effective way of helping them to complete their task.
There's nothing they're more close to than their cell phones.
Yeah.
If you can get something off their cell phone, it's theirs. It's they can possess it. They can connect to it somehow better than if it's not. And you prepared them by drawing all these things on the board so they knew what to look for, more or less. And,
and I spent about 20, 30 minutes actually making some detailed drawings of some of it, because it was really fascinating. Those rotifers, and they're moving their cilia and they're pulling in the filament algae and they're eating it, and it's really cool. You can even see the digestive system.
Yeah.
Because they're pretty much clear, clear little critters.
So they're actually seeing all sorts of parts inside cells.
Yeah.
Or in the case of the rotifers, this is a multicellular thing. I mean, you can see the grinding parts in the throat. You can see a little pulsating heart.
which is great because we did digestion and we did cells, and we're going to be going into next month or maybe after the break, we'll be going into the whole human physiology. So we'll be studying respiratory, digestion, everything. So I think it's good that they can actually see digestive system happening
Yep.
live.
Yep. and the whole little ecosystem right there that just gets them. We can always use this as a reference when we start talking about the food chain, food web, and the algae, the blue-green algae. You can always make reference. And the detritus, the dead material, that's being fished through by all kinds of things, including even nematodes and earthworm relatives who are squirming through some of that. So the idea that detritus is an important part of the food chain and working that back into recycling minerals, recycling materials, and breaking it down as decomposers. But they're looking at a whole mini ecosystem here, and then they can apply that on larger scales also. So this has some spin-offs that are all really useful and helpful, and we also get a sense of the size of cells. We've done some measuring using rulers. We've measured the 4 millimeters across at the low power, 2 millimeters across at medium, and 0.6 I guess it was. So we can actually get a guesstimate of the size of these things. So some of which you can see actually if under perfect conditions, the larger things. So they start to get a sense of size and realize this stuff is just below, a lot of it's just below what they can see for the most part. But some of it is just bridges that gap slightly, and they can just barely see these things. So all good stuff.
I think because it was a group project, what I did when I picked the groups up, I didn't let all the best friends hang out together. I split them up a little bit. So I put some of the top students, made sure there was 1 or 2 top students in each group to kind of bring up the level of learning with the students that are having more challenges.
Yeah.
And some of the girls, especially, their drawings are so perfect. But if the other kids can kind of look and see what they're doing, it can help them to do that. And also just the microscope skills and everything. There's a difference in some of these kids because they're high school kids. Some kids are more mature than others.
Yeah.
Some kids get it faster. So I was careful when I let them collaborate that I kind of mixed the groups so that they would all have some help.
Yeah, great. And a lot of techniques used there. You had to stain the cheek cells. Amazing that you can scrape the inside of your cheek, and then next thing you know, you're looking at cheek cells stained and cover slip. And use of the technique, appreciation of how a microscope works, how to be protective of the scope and not use force focus on high power.
Break the lens.
Yeah, how sensitive it is, and that the lenses have to be carefully wiped off with special paper, lens paper, not any paper, these kinds of things, so we can still be able to see by the end of the year. Let's see, I don't know, what else was I going to say? I was going to ask you about improvement. How do you think you could improve this thing?
Well, I think that my knowledge is not as great about all the different little critters that we were looking at.
Not too many people spend a lot of time.
Yeah, but I know anatomy and physiology of the human really well. But I think if I could have read more and learned more about them, I could explain it better to the kids. So I think we did the lab. We had a project right before that, and this lab came up, and I don't think I prepared as well as I could have. I think I could have prepared better.
Okay, so more info about the critters,
what we were looking at.
Yep. Pointing out things like euglena, which it kind of blows away your idea of animal and plant, and the classification. What are these things? Are they bacteria? Are they not? So no, most of them at least belong to a whole other kingdom. of simple-bodied, single-celled, and all simple-bodied things, including the algae and protozoans, that we call the protista. Not all, there's some exceptions to that, but a lot of them are in that group. The misconceptions, do you have any that you cleared up, do you think, from all
Well,
this? Or?
I think that a lot of the students were just very um, taken back and amazed that we could just go get some mud puddle water and there was life in there. I'm not sure that they were aware that that existed, that whole world existed. There's a whole ecosystem that that normally they would just walk through it. It just came from the rain. There's rain and dirt, and kind of we're in a developing country, so the streets are not exactly clean.
No,
it's a gross understatement. And anyways, so I think the kids were very amazed to see the life. And normally they would just look at that water and think it's just runoff water.
Yeah, the monsoon, does, even though as long as it keeps raining, usually it doesn't get too dirty. There's enough flushing that you get relatively good conditions. One of the places we collected, it was green. The water actually was green, and so to be able to see why it's green, this is a pretty cool thing for them to see. But it's amazing how similar all over the world this stuff is. It's not. I mean, this puddle could have been anywhere. It's similar kinds of things you see, whether it's Burma or New York or whatever. So there's a lot of similarity on a global scale to at the microscopic level. Yeah.
Yeah, I think it was a great lab. I think the kids got a lot out of it. They really enjoyed the lab. So much of our teaching is we're showing animated videos. We're talking about photosynthesis. We're talking about the Krebs cycle, electron transfer chain, and those things they're interesting and they're very important to learn in biology. But this was kind of something they could really get their hands on because they could actually see life. Yeah. And I think it was a great lab.
And it moves.
Yeah, I think it was a great lab.
They can move and it moves.
Yeah.
They love that stuff. Anything that's moving kind of grabs their, especially when it zooms across, it gets them excited. And now the next level would be for them to be able to chase these things down. But usually what you have to do is either put some sort of something to thicken the liquid, slow them down, or let it dry up a bit so it restricts where they can go, and then you're able to follow them better. They have less room to roam. So that's always interesting as they dry up with it towards the end of class. Just they're kind of more restricted, and they can really see some of these things. They kind of get stuck and they spin around or they stay in place. Then you can really see inside the guts of them.
Yeah, but I think overall it was a great lab because they got to open up a whole new world to them, a whole new environment, ecosystem, and they used technology because they limit the number of limited microscopes. Yeah, you got to practice using the technology. So I think it was for the time that we had, I think it was time well spent with the kids.
I do too. I guess we covered most of what we could cover.